Coming up ahead in this episode of Xtalks Spotlight.
And so having an assay that can provide a same-day result in just a few hours to identify and quantify all of the mRNA constructs that are in that multivalent mRNA vaccine mixture can absolutely help speed up development timelines in process testing and reduce QC bottlenecks.
Hello, and welcome to Xtalks Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we're asking the question, how can rapid mRNA quantification enhance multivalent vaccine development?
The surge in mRNA vaccine candidates has driven the need for faster, more precise analytical methods. As researchers push the boundaries of mRNA-based medicine, optimizing techniques for identity confirmation and quantification is essential, especially for multivalent vaccine development. In this Xtalks Spotlight edition, we spoke with Dr. Erica Dawson, Chief Research and Development Officer at InDevR, who shared insights on innovative analytical approaches for addressing these challenges. Dr. Dawson explored how rapid methods are setting a new benchmark for efficiently identifying and measuring multivalent mRNA constructs. Thank you for taking the time in the Spotlight interview, Erica.
You're so very welcome. Looking forward to talking to you.
So to start us off, could you share an overview of the recent developments in mRNA analytics and how they are driving advances in multivalent vaccine development?
You know, it's interesting, but that's just it. There isn't a lot of emphasis right now being put on new method development specifically for application to multivalent mRNA vaccines. And I think this is surprising given just how many multivalent mRNA vaccines are currently in clinical development, right? So not only combination ones like influenza and COVID, but also just seasonal influenza, RSV, other combination vaccines. And, you know, there have been some recent reviews in the literature highlighting improvements in technologies like mass spec-based methods for doing mRNA analytics, but these aren't necessarily with an eye toward multivalent vaccines. And so that some might say that doesn't seem problematic for assessing each individual drug substance in the overall vaccine. But when it comes to final QC release tests in which the drug product is already formulated and all of those mRNA constructs are already mixed together, many of these existing methods are going to become a challenge. And so because these methods will struggle either in their ability to measure multiple constructs independently or being non-ideal from a need for upfront sample prep or sample processing, such as like requiring an upfront extraction of the mRNA from a lipid matrix, or they could be complicated from an expertise or a facilities standpoint. So many of the current methods involve sequencing. And so there's a lot of upfront sample prep, a lot of complex back-end data analysis and bioinformatics. And one of the other types of methods that's currently utilized are RT-PCR amplification-based methods. And those can handle a specific amount of multiplexing, but they do require upfront extraction of the mRNA prior to amplification. And of course, that needs to be done in sort of a specialized nuclease-free environment and requires a certain amount of environmental controls to be, for that process to stay contamination-free. So while there certainly have been advancements in mRNA analytics, really, there hasn't been a whole lot of advancement with an eye towards the increasing amount of multiplexed vaccines that are in the clinical pipeline.
And what would you say are the main benefits of being able to confirm identity and quantify multiple mRNA constructs in under 2 hours?
I mean, it sounds cliche, but time is money, right? For pharmaceutical companies and vaccine development companies, time is money. And so having an assay that can provide a same-day result in just a few hours to identify and quantify all of the mRNA constructs that are in that multivalent mRNA vaccine mixture can absolutely help speed up development timelines in process testing and reduce QC bottlenecks. And so, many of the large pharma partners that we've talked to mention that oftentimes the current methodologies, they have to outsource. That could be internal outsourcing to a centralized analytical lab that happens during bioprocess development, or to a central, like a completely external lab, like a CDMO or something like that, because they require specialized scientific expertise to actually execute these analyses. And in addition to that, often samples end up sitting in a queue prior to analysis, sometimes up to weeks before they actually get analyzed. And so, by definition, that waiting sort of delays any actionable information received from those analytics that could help inform the bioprocess, which ultimately then can delay the development time. But aside from just time to result, another really important aspect of adoption of a new methodology is to make sure that the assays have high ease of use, good performance in terms of their repeatability between users. And now that we live in such a global world, not just reproducibility between users, but also between sites. Given the global nature of vaccine manufacturing, often these same assays are having to be performed at multiple sites across the globe and run in different laboratories. And so I think really these are some of the key benefits of being able to confirm identity and quantity. It's really to have same day actionable results in the bioprocess lab or in the QC lab to really speed up development times.
Now, when it comes to accurately quantifying multiple influenza HA mRNA constructs, which are key to developing effective seasonal vaccines, what have been some of the major challenges and breakthroughs that you have seen?
InDevR had funding from an organization called Nimble here in the US, and they asked us specifically to develop a identity and quantity assay that could be universally used to quantify all of the hemagglutinin coding constructs that would be in a typical seasonal influenza mRNA vaccine. And so I think really the key challenges here are the nature of influenza, right? First, the strains that are recommended to go into a seasonal influenza vaccine can change annually, right? Because of the ongoing antigenic drift of the virus, that's obviously, this is kind of a moving target. And also, because that protein sequence of the vaccine strain changes, that by definition means that the mRNA construct sequence will then need to change in lockstep with that in order to code for the correct protein. And so one additional challenge is that, above and beyond the sort of seasonal drift of the influenza virus, everyone who's going to manufacture an influenza mRNA vaccine is very likely to use a different proprietary codon optimization scheme to choose which codon get used in the mRNA construct to code for each amino acid in the protein that gets made. And so even constructs that code for the same protein ultimately can have very different sequences at the mRNA level. And so really the way that we achieved universality was through two things. First, careful sequence design of the capture oligonucleotides on the assay that we developed. And second, careful optimization of the hybridization and wash solution formulations to optimize the performance of the assay. And so in terms of sequence design, we did essentially amino acid sequence alignments of all of the recent influenza vaccine strains for each component in the vaccine. And then we assessed that amino acid space for areas of sort of sequence similarity for related viruses. But we also had to focus on trying to determine which stretches of those amino acids were going to be less subject to codon optimization. So where there was less possibility to have to choose different codons in a certain region. Again, while still needing to provide specificity against the other components that are in the vaccine, right? So, there are 2 influenza A's and now 1 influenza B in the current recommendations in terms of seasonal viruses. So we have these 3 challenges really of having to be specific against the other vaccine components and then also having to be more generally universal to viruses as they mutate over time for a specific component, and to be robust against those different codon optimization strategies. And so we do that not only through sequence design, but also because nucleic acid hybridizations are very sensitive to the salt conditions and the additives that you have during the hybridization event, we also need to balance these 2 sort of competing properties of specificity and sensitivity or reactivity, right? So still getting good, strong binding, but maintaining the specificity and not getting any off-target hybridization that would be unwanted, that would convolute the results. And so thankfully, We did all of this work, and we now offer these in sort of a prepackaged, off-the-shelf reagent kit that can be just used as is and tested on a wide variety of different influenza HA mRNA vaccine constructs. So really a technical challenge to make this kind of assay universal.
So you just mentioned working with Nimble, and I know that you also had a recent collaboration with Pfizer on custom assays. I would love to hear any lessons learned that might shape the future of mRNA vaccine development.
Given that we develop tools for the vaccine manufacturing industry and not vaccines themselves, I may not be the most well poised to talk about the factors driving sort of future vaccine development. But what I will say is, there are, of course, a variety of different multivalent mRNA vaccines in clinical development. And this need for better multiplexing analytical tools is not going away. I might also point to the increase in vaccine hesitancy that we've seen post-pandemic, and that increasing the valency of vaccines through strategic combinations will ultimately help reduce the overall number of vaccine administration events, right? Minimize the number of shots in the arm someone needs to get. And so as these multivalent mRNA vaccines become more commonplace, certainly the burden of the analytical methods used to assess them is going to go up. Specifically, the one comment I will make in terms of the joint work with Pfizer was that work was based on very specific mRNA construct sequences that were provided to us by Pfizer for a vaccine that was in clinical development. And so that development was actually technically far more straightforward than developing a universal assay like the one that I just talked about. And that's because there's really no requirement for the assay to be forward compatible with other construct sequences as a function of time. It was developed for that very specific formulation. And I think what that does is help, InDevR is in the business of really helping vaccine manufacturers try to streamline their analytics. And I think what we learned from Pfizer was that it was actually technically quite straightforward for us to develop that assay, and that the timeline for a custom assay to be developed from start to finish was actually quite rapid, on the order of just several months, in order for us to develop and validate a fully off-the-shelf assay for the purposes of identity and quantity for a multivalent mRNA vaccine.
So let's wrap up by talking about the evolution of mRNA therapeutics. How do you see mRNA analytics moving forward to support multivalent vaccine development? And what can we do today to get ready for these future challenges?
I'll come back to my answer to the first question in that, there really hasn't been a lot of in-depth analytical development with an eye towards multivalency, right? And a lot of the current methodologies that are used to analyze mRNA constructs end up sort of falling apart as you add additional constructs into the mix. And so one thing I hope we will see is the advancement of mRNA analytics that are more fit for purpose. And so an example I will give is the use of sequencing to do routine identity testing, right? So identity testing where you have to essentially confirm that the construct that you say is present is indeed present. I will not argue that, understandably, early in a program's development, you absolutely may need a tool like sequencing to understand every single base in the entire mRNA construct, to indeed make sure that you are producing the full construct that you intend to be producing, all the way from 5' cap through the 5' untranslated region, the entire coding region, the 3' UTR, and ultimately the 3' poly A tail, right? There are parts in the program's development where you must know that every single base that you expect to be present is there. However, once the process of making your mRNA construct is fairly understood in a program's development cycle, I would question whether or not it still makes sense to use that high information content but complex technology like sequencing to do routine in-process identity testing. This, in my opinion, seems a little bit like using a sledgehammer to crack a nut. It's a bit overkill for the task at hand. And I would say the same thing about final release testing in QC for identity as well. You've now been working with these same constructs all along during your program's development, And now, really, you just want to confirm that you have all of the components in the vial that you intend that are going to go into someone's arm. And so my hope moving forward is that there's going to be increased acceptance both by the manufacturers and by the regulators for in-process testing and QC testing applications that may be a bit lower information content, like the VaxArray approach that InDevR offers, but that have the benefits of being provided in a pre-packaged off-the-shelf kit that enables good reproducibility and standardization across sites, are fast to conduct, and so give quick, actionable information and don't require any complicated upfront sample pre-processing.
Well, thank you so much, Erica, for speaking with us today. We really appreciate your time and insights.
Thanks so much for your time, and I really enjoyed it.
We look forward to hearing more about InDevR's efforts in making multivalent mRNA analysis faster and more efficient than ever. Thank you all for joining us for this Xtalks Spotlight feature. We hope you enjoyed the discussion.